Hello students, today we are continuing the yesterday's part. So quickly I can revise the yesterday's topic, what we discussed in our yesterday's class. That is when we take the whole earth, I mean to say the whole locality is considered as earth. Within that earth, a small part, a small unit is called as biosphere. So within the biosphere we will be having living system, non-living system, yes or no? So in that, I mean to say that according to this topic, that is biomolecules, yes or no? In that, the molecules which are present in the earth crust, I mean to say that we can call it as a lithosphere. The same time, those molecules which are present in the living system, are they similar? So that question mark that leads to the whole chapter. Yes. So to understand what are the different chemicals present in the living system and non-living system, I mean to say that one such example is carbon. Yes. So the carbon is present in the non-living system as well as in the living system. So the same carbon is present in the same, I mean to say in the soil as well as in the living system. Are they similar? Yes or no? That's a similar. So when we understand the answer, there we can understand, that is, so the carbon which is present in the living system and in the soil or in the non-living system, say their carbon will be remain same, but their functional aspect will be differ. So to understand this concept, we understood an experiment called as chemical analysis by taking biological sample, mortar, pestle, yes or no, by addition of trichloroacetic acid. After complete that chemical analysis experiment, the scientists have discovered thousands of different organic compounds in the filtrate or in the acid soluble fraction. And one more is that is a destructive experiment. In the destructive experiment, same, we need to take biological sample and try to allow it, allow it to dry, and after complete removal of water, try to burn that biological sample. So in that we are going to get two fraction. One will be all carbon compounds can be oxidized in the form of water vapor and carbon dioxide, and one more, that is what we get after burning, that is ash. So that ash contain inorganic elements, inorganic molecules and inorganic compounds, clear? Such as calcium, magnesium, manganese, yes or no, and sulfate and phosphate. These are the what, some inorganic elements as well as compounds, clear? And today we are going to continue the yesterday's part, that is, see, why we are conducting these experiments? To understand what are the different chemicals present in a given biological sample. Yes, so we have some various separating techniques. I told you yesterday, that is chromatography, thin layer chromatography, paper chromatography, gas chromatography. We have different techniques. So by means of those different techniques, definitely we can categorize, we can separate the various chemicals which are present in the biological sample. Yes, so and this chemical analysis, that is elemental analysis. Elemental analysis can provide you the chemical composition. Do remember this point. Yes, the elemental analysis, see, it may be chemical analysis experiment or it may be destructive experiment. So both can provide the chemical composition. I mean to say what are the different chemicals present in a given sample, that is given biological sample. This is most important, clear? So after completing this one, we need to understand their chemical structure as well as chemical formula. I told you yesterday, chemical structure as well as chemical formula. After this, whatever the chemicals we got, do remember, very important point, whatever the chemicals we got from the chemical analysis experiment as well as inorganic experiment. So here when we give that chemical, clear, it may be organic, it may be inorganic, it may be inorganic element, it may be inorganic molecule, or it may be inorganic compound. So whatever it may be, so those all chemicals, when we give those chemicals to the chemistry people, do remember, chemist, when you give these chemicals to the chemistry individual or chemistry a scientist, a chemist, so definitely he is going to give, I mean to say that according to chemistry point of view. Do remember, whatever the chemicals we have right now, so those chemicals according to the chemistry point of view, so he is going to understand their functional group. Please do remember this. He always bother about, we always concern with what are the different functional groups are present in a given chemical, such as we know that some of the examples I can write to people, that is ketone. As we know that one will be what? Aldehyde group. Aldehyde group. One will be carboxyl group. Yes or no? Another one will be what? Hydroxyl group, that is OH group. So that what I am trying to tell you is that when you give whatever the chemicals we have to the chemistry people or chemist, so according to the chemistry point of view, the chemistry individuals always concerned with the what are the different functional groups are present, such as ketone, C double bond O, aldehyde, COH, carboxyl group, that is COH, then hydroxyl group, that is OH group. So these are the different functional groups attached with the main carbon. Yes, this is what according to the chemistry. The same time, when we have a chemical, so when we understand or when we give this chemical to the what? The biological individuals, or we can call it as biologist, scientist. So according to the biological point of view, he always concerned with a very important word, that is functional aspect. Do remember, he always concerned with the functional aspect. I mean to say that what it does in our body, what it does in the living system, what it does in the plant body or whatever the living systems are there, so that he always concerned with the functional aspect. Along with that, he also concerned with, that is according to the biological point of view, he also concerned with, that is whether those chemicals belongs to, do remember, he always concerned with, one will be functional aspect, second one is, that is those chemicals belongs to amino acids, I mean to say the protein, or they are nucleotides, I mean to say which are present in the DNA and RNA, like we can call it as nucleic acids. The same time, whether they are belongs to the carbohydrates, yes, and one more is belongs to lipids. Yes, so there are two aspects from the biologist. One is, that is functional aspect, second one is, so whether those given chemicals belongs to amino acids, I mean to say that proteins, or they belongs to nucleotides called as nucleic acids, that is DNA and RNA, whether they are belongs to the carbohydrates and lipids, simply called as what? These all simply consider as biomolecules. Do remember, these simply consider as biomolecules. I hope you understand. At the same time, if you give these chemicals to the physicist or physics, what happens? Can you understand? Yes, he always concerned with, I mean to say the physics individuals always concerned with the chemicals, that is their physical dimension. Yes, dimension. Say for example, imagine is a kind of chemical. Here will be having a chemical, a carbon atom. Here will be having a what? A functional group. So if you hold like this, you are going to observe this face is considered as one dimension. I can hold like this. Same time, meanwhile, you people are going to understand this face as well as this face is considered as two dimension. I can do like this. So one phase, second phase, third phase, that is third dimension or three dimensional structure. Yes or no? So likewise, they are going to understand, that is physics. You must remember this. One is what I mean to say, whatever the chemicals you are going to provide to the chemistry, biology and physics. Chemistry, according to the chemistry point of view, they always concern with a functional group. Biology, functional aspect. Functional group is different from the functional aspect. What it does in the body, consider as functional aspect. Then belongs to those different chemicals, whatever we are providing, those chemicals belongs to whether they are amino acids, nothing but proteins. Nothing but proteins. Second one is nucleotides and carbohydrates and lipids. So this is what according to the biological point of view. And last one is physics. They always bother about the dimension. What are different dimensions are present in the given chemical. This is one aspect. Clear? At the same time, we will move on to the next one, that is amino acids. Next topic, small topic, is amino acids. Next, what is amino acids? Very simple. When we have a protein, I mean to say that when we have a protein molecule, we can call it as a complex food molecule. See, complex food molecule. So why I am using the word complex food molecule for the protein? Because protein is a one such molecule which is made up of several thousands of, the protein is made up of several thousands of small units. Those small units, each unit is considered as amino acids. Do remember. Clear this? So I mean to say, what is protein? Protein is a long chain of amino acids. Clear? Now what are amino acids? As I told you right now. So when the protein undergo series of metabolic process, finally it leads to become, that is amino acids. So for that reason, what are amino acids? Amino acids are the end product of protein metabolism. Very simple. Once again, what are proteins? Proteins are the long chain of or polymer of amino acids. In the same time, what are amino acids? Amino acids are the end product of protein metabolism. When protein undergoes series of metabolic process, leads to formation of amino acids. So this is what the different the definitions or the explanation for the protein as well as amino acids. Now what are amino acids? Amino acids nothing but these are the end product of protein metabolism. Clear this? This is one topic in this. So when we understand the amino acids, these all belongs to the organic compounds. First point. These all belongs to organic compounds. Yesterday I told you people while discussing about chemical analysis experiment. So we had a test tube. Test tube was covered with a cheesecloth or cotton. So when we pour the content from the mortar, which contain biological sample plus Cl3CCOOH, yes or no? So after that we will be having two fraction. One is retentate or acid insoluble fraction, and second one is here we will be having filtrate. We can call it as acid soluble fraction. So I told you yesterday, scientists have discovered thousands of organic compounds in the filtrate or in the acid soluble fraction. So in that one such, that is amino acids. Yes or no? Because I told you yesterday, here you will be having thousands of organic compounds. So those organic compounds nothing but the proteins. Clear? Nothing but amino acid. This is one thing. Next, the very important thing to understand about the amino acids is, so when you read the word amino acid very carefully, there you are going to understand two things. One is, amino acids are the one such molecules which contain amino group. Do remember, one will be amino group, that is CH2. Yes or no? CH2, that is amino group. At the same time it will be contain an acidic group. Yes or no? One will be acidic group, that is COOH. Do remember. I mean to say that a single carbon, here you will be having a single carbon, that single carbon contain both basic group, that is NH2. At the same time, same carbon contain an acidic group, that is carboxylic acid. Yes or no? Carboxylic acid. So that this kind of amino acids are called as alpha amino acids. In the CT examination, they are going to ask, do remember what are alpha amino acids? The amino acids, a single carbon which contain both amino group, that is basic group, and other side it will be having acidic group. One will be basic group, another will be acidic group. So such amino acids are called as alpha amino acids. Very clear? At the same time, when you talk about this amino acid, one more very important thing is, see, when we have a carbon at the center, here we will be having, I told you right now, COOH, that is carboxylic group, or we can call it simply called as acidic group. At the same time, here we will be having NH2 group, yes or no? NH2 group, and here we will be having one hydrogen group and one will be R group. So this is what the typical structure of amino acid. The typical structure of amino acid. Here the we need to understand is, here one is acidic group, one will be basic group, one will be hydrogen. Yes or no? Hydrogen. At the same time, here we can call it as variable group. We can call it as variable group. So the word variable group refers to oftenly it can change. Oftenly it can change. Hence it is called as variable group, or we can call it as aryl group. R is also called as aryl group. I mean to say that this is what I should not call by any particular amino acid. So simply consider as typical amino acid. So I mean to say, particularly if I use any particular amino acid, means this R is going to change. This R is going to change. So what kind of group is present in the R? So based on that, we are going to name the different amino acids. Different amino acids. Here till now, till date, the scientists have discovered 20 different amino acids. Do remember. Till now, our scientists have discovered 20 different amino acids. Clear this? So, some of the examples I can give you people: alanine, arginine, aspartic acid, glutamic acid, glutamate, then tryptophan, valine, serine, so glycine. There are various different types of amino acids are there in the nature. I mean to say, so totally there are only 20 amino acids. But according to one study, I can tell you people, so recently scientists have discovered 2 more amino acids, but still they are not included in the 20 amino acids. Do remember, so those 2 are still, about to put, but still they are not included in these 20 amino acids. But according to NCERT book, there are 20 amino acids. At the same time, one more thing I can tell you people, I told you the name of the amino acid is going to change according to the aryl group. So one such example is that if I put hydrogen in the place of R, I mean to say that carbon, COH, here NH2, and here hydrogen, one more hydrogen I can put in the place of R. So definitely it will become glycine. Do remember, it will become glycine. Glycine is one of the amino acid out of 20 amino acids. Clear? This is how. And second example I can give you, if I put methyl, that is CH3, see, COH, here NH2, and here as it is H, here I am going to write CH3. It will become what? Alanine. It will become alanine. If I put CH2OH, that is hydroxymethyl, it will become hydroxymethyl, like what? COH, again NH2, here H, here CH2OH. It will become serine or serine. Do remember. So this is how the name of the amino acid depends on the R group, that is aryl group. Clear this? This is how we need to name the different amino acids. Very clear? Very clear. Yes. Next. Next, based on the, here I told you many times, basic group, acidic group. So based on the number of carboxyl group and basic group, that is amino group. So we are going to write some different amino acids, that is acidic amino acids. Acidic amino acids means no doubt the amino acid which contain both acidic group as well as basic group, but thing is more number of acidic group, more number of carboxyl group, so we can call it as acidic amino acid. So example, as the name indicates itself, glutamic acid. Glutamic acid is one of the amino acid in 20 amino acids. Is acidic amino acids, and one will be basic amino acids, means same structure but the number of this one will be higher than the acidic group, so consider as basic amino acids. Example, alanine. Is alanine, do remember. Is next one, we will be having neutral amino acids, nothing but the number of basic and acidic will be same, so consider as neutral amino acid. Example, valine. Example, valine. At the same time, you will be having some aromatic amino acids, some aromatic amino acids. So here aromatic amino acids, example, tryptophan. Then we will be having what? Tryptophan. Best example is tryptophan. Apart from this, we will be having still many are there. That is what? Alanine is there. So leave it. Aromatic amino acid. Best example is tryptophan. So these are the what? Some different amino acids according to how the number of acidic and basic group will be present in the respective amino acid. Clear? So this is what today's topic. And one more last topic is zwitter ionic. So sometimes the amino acids play a different role based on the nature, what kind of solution is present in the given medium. So for example, I can write, that is carbon, here you will be having COH, here you will be having NH3 plus, and here you will be having H, and here you will be having R. And here imagine it is A. Same time it will become is one hydrogen is going to remove, it will become COO minus, as it is NH3 plus and H, and it is R. Imagine it is B structure. The same time here it will become C, same time CO minus and aryl group and hydrogen group. Here it is going to change like, that is NH2 minus. It will become NH2 minus or NH2, simply NH2. Clear? The same time, when we add hydrogen to this, when you remove hydrogen, it will become NH2 minus. When you remove one more hydrogen, it will become like this. Yes or no? Same time, when we add one hydrogen to this, it will become like this. Yes, that is going to get one hydrogen. Again, when we add one more hydrogen to this one, it will become NH3 plus plus COH as it is. So called as zwitter ionic form. This is also one of the nature of amino acids. So this is enough for today's class, and remaining things we'll discuss in tomorrow's class. Thank you.